Endocytosis is a form of active transportation, or way that a cell is able to take in nutrients and particles. Endocytosis is able to take in nutrients by surrounding nutrients with the cell membrane and bringing that nutrients inside the cell. This process is used to gain nutrients and can also be used to remove parts of dead cells from extracellular space. There are three types of endocytosis: phagocytosis, Pinocytosis, and receptor-mediated endocytosis. The first process is phagocytosis. This process is when the cell membrane will surround one large particle. This particle will then be taken into the cell to be digested. The process starts when projections of the cell membrane are sent out to surround the particle. These projections will then combine to fully engulf the particle. When the particle is engulfed, it is inside a vesicle called a phagosome. The phagosome will be attached to the lysosome, where they will be digested and broken down into smaller parts that will be used by the cell later. Phagocytosis is most important for its role in the immune system. It is used to remove pathogens. Although phagocytosis is mainly used to remove disease in some organism, it is also used to gain nutrients. The second process is pinocytosis. This process happens in areas of the cell membrane where pockets can be formed. These pockets are filled, then pinched off, and form vesicles. These vesicles are filled with extracellular fluids with particles inside the fluid. The pocket is filled without a filter on what can enter. The pocket will then fuse with other cell structures to be filtered. There is also an opposite process called exocytosis. This process will excrete unuseful particles and add back to the cell membrane, causing a cycle with the two processes balancing each other out. The exocytosis will only send out non-useful particles, letting some particles be filtered out of the cell. Because pinocytosis is unable to filter the particle coming into the cell, because phagocytosis is unable to filter the particles coming into the cell, it is the least efficient. However, phagocytosis uses the least amount of energy, so it is still used by some organisms. The third process is receptor-mediated endocytosis. This process starts with multiple receptors which are attached to the cell membrane. These receptors are sticking out of the cell and will stick to a certain particle. They will only attach to that specific molecule. After a molecule is attached to the receptor, the plasma membrane will make an inlet for the receptors to be pulled into. The membrane will then pinch off the inlet and form a vesicle. This type of endocytosis is most commonly used type due to its ability to collect certain molecules, even though it's able to select specifically. It's not used in all organisms because the process is not effective. Phagocytosis, pinocytosis, and receptor-mediated endocytosis are all ways a cell can take in molecules. Each method has its own benefits and downsides, but each method is important to a certain molecule